Okay, let's get started. We'll catch people up as they join. Thanks for joining our webinar today. Today we're going to talk over how to use the Cube Lab CRD Botrytis test. So a couple housekeeping issues. We're going to try and keep this to around 20 minutes of presentation and then open it up for questions. If you look at the top left, you should see a Q&A box, and you can put down your questions there. We can either answer you directly or after the presentation, we'll review all those questions. Thanks again for joining. So what is the CRD Botrytis test? We are using lateral flow technology, which now just about everybody's used one of the new COVID tests. We use antibodies and antigen reactions to determine levels of pathogens in the field. So some of the reasons we like this method is it's very reliable, very accurate, very sensitive. You can get a good spread of testing from across your field, as well as a low relative cost to sending off samples to the laboratory. So this is the antibody that's used on the test strips. And again, it's very, very sensitive to the entire Botrytis genus. It doesn't really react with other pathogens. So, if you have Fusarium or any other nasties in the field, you can help diagnose it with our test and make sure you know what you have in the field. It's very accurate. So what we did is take a reading using our test compared to known quantities in a solution. It's very comparable to things like qPCRs, LAMP assays. It is fully quantifiable. So if you have a very low level, it shows up as a light line, or a very dark line if it's a high level. So now that we know exactly what we're dealing with here, there's a few methods that we can use in the field to help the farmer make decisions. So the first one is spore detection, or what we call early detection. That helps you measure the spore levels before infections are visible or even infected, so you can address your treatments accordingly. If you need to diagnose what's happening in the field, you can do an immediate test and get your Botrytis levels right then and there. We can help you validate your treatments and avoid resistance, save on sprays, target sprays, some really exciting things you can do with the test on that level. And then post-harvest quality control. So managing your risk as the product enters the supply chain. I'm going to show you a quick video here, swap screens, and show you just how easy it is to use the test.
Today we're going to show you how easy it is to use the Cube Labs Rapid Pathogen Test. Step 1, sample prep. You can sample the flower, leaf, stems, roots, or the fruit of your plants. Be sure to test both healthy plants as well as any that are showing signs of disease. The whole sample should be the size of a penny. Add the sample into the white capped bottle along with 3 BBs. Return the white and green caps and ensure they are snug. Shake the bottle for 15 to 30 seconds to break up the sample. Step 2, early detection. An early detection test is used to detect spores and early stage infections. To perform an early detection test, add 1 germination pad into the sample bottle and leave it in a dark place for 12 to 24 hours before testing. Step 3, running the test. Take out a new test strip and test well. Make sure to reseal the test strip container. Remove the green cap from your sample bottle and squeeze 4 drops into the test well. Next, add 1 drop from the black capped test activator bottle to the test well. Hold the test strip by the red handle and place the blue end into the test well. Let the test run upright for exactly 10 minutes. Step 4, reading the test. After 10 minutes, remove the test strip and read it using the guide on the instruction sheet. The top control line indicates that the test worked. The lower sample line indicates if a pathogen is found. The darker the line, the higher the pathogen level. Thanks for learning about using our rapid pathogen test. If you have any questions or would like to learn more, please visit cubedlabs.com.
Okay, so as you can see, if you've run pH test strips or some of the similar things and used on the farm, you don't need much training and you can get really usable results using our test. So first thing we want to speak to is how to set up a testing protocol. And we can do this whether you're indoor farming or outdoor, greenhouse, field grown. But it's important to break up your field so that you really get a good understanding of what's going on. Some of the key factors to consider in sample collection is high traffic areas or wind direction. So, on this farm, there's a road right here, dust and debris is going to be falling into this plot of land. So we're going to take an extra sample from closer to the roadside. Pathways, ventilation systems in your greenhouse or indoor growing facility. Another thing to consider is existing neighboring foliage. We've found botrytis infection coming in from neighboring wooded areas. If you have neighboring farms, if you're a strawberry farmer next to a grape vineyard, there's a high risk of cross-contamination back and forth. So you really want to test around the perimeters. There's also different microclimates to consider, wind direction. You can just take a couple extra samples. These yellow dots taken over about an acre of land in a greenhouse, 2,000 to 3,000 square feet of space. Similar grid can be set up. And it really gives you a detailed map of what's going on, where the infection's coming from, and how you can manage that a little bit better. We also recommend piggybacking our test onto your current IPM scouting protocols and programs. So if you have scouts in the field already or labor working and they see something in the field, all they have to do is have one of our sample tubes in their pocket. They can take a sample, test it, and it helps you diagnose on the spot. One thing, if you would like to use our test kits, we're more than happy to get on a call with you, take a look at your property, and help you set up a grid system like this if you don't have that already. So please contact us and we'd be more than happy to help set up some of these protocols for you. So first method that we like to use, and this is the primary use for our test, is in early detection. So it's quite simple. All you have to do is collect the samples. That can either be a tissue sample, a fruit sample. We even have people in greenhouse and indoor facilities take swabs of environmentals, with HVAC systems or fans. They always get dusty. You can swab it with a Q-tip or a sterile cotton swab and use that as your sample and put that into the buffer tube as well. So once you have your sample collected, you need to add the germination tab. And in the kit, it comes with 2 bottles. I'll grab them right here so you can see. There's two bottles. One is labeled germination pads and one is labeled test strips. So you want to take out that germination pad. You want to take out the germination pad, take one of the little pieces of paper out, add that in with your sample, close it back up, make sure the lid's nice and tight so you don't spill it. And then we run a germination stage, and that takes about 12 to 24 hours. What we're doing there is allowing the perfect environment for those spores to germinate. That germination pad gives them a little bit of food so that the mycelium can grow out, and that's when we can test for it. So after the 12 to 24 hours, we run the test as you saw in that video, and we'll get into a little bit more detail here in a minute. The results for this are very important, and again, we recommend this is the highest use case for our test to know what's in your field, what the risk factor is of you having an outbreak. So if you know the spore count and you monitor the weather systems coming around, we all know that botrytis and a lot of other fungal pathogens require specific humidity ranges, rain events, or temperature ranges. So if you know your risk of spore pressure and compare that to the weather systems or your environment in the greenhouse or indoor facility, you can really get a good map of what your risk profile is. So the next method of use is the instant test. This is really great for diagnosing in the field. So similar to the previous method, you collect your sample. Again, we don't recommend roots and soil. It can tend to clog up the test strip. So keep it to stems, leaves, fruit, or environmentals. The second step is you add the little BBs provided in the kit and get to shaking. And that helps break up the plant material so that you can get access to the internal infections if they're present. Shake it for 30 to 30 seconds around there. Add 4 drops of the sample dropper, the green top dropper, to the test well. And then you have to add 1 drop from the black top test activator bottle. And that helps the test run accurately. Once you've got the liquid in the test cup, you add the test strip, and it's just a waiting game. So it takes about 10 minutes for full results. You typically see, if you have an active infection, you'll see the test line start developing much sooner than 10 minutes, but we recommend you wait for 10 minutes to get the full result and then remove the test strip from the test cup, and you can read the results. On the instruction sheet that comes in the test kit, on the side here, you'll see a barcode or a graph that shows you infection levels. So the lighter the line, a light line, or it kind of looks gray, that is typically a low level of infection, but still needs to be addressed. And a red line means take immediate action. You have an active infection. The next method is in treatment validation. So we really are seeing some good results on this. You need to work with your agronomist, your PCA, whoever, whoever helps manage your treatments. After your spray or your treatment, whether you're using synthetics or biological controls, take about 3 to 5 days and go out into the field and start taking some samples again. So what we recommend is after you've treated, you take your samples and you run an immediate test. That gives you the baseline of infection. So our test does detect dead mycelium of botrytis. So you can get that measure, compare it to the chart, say, hey, I have an infection level 3. Let it incubate for 12 to 24 hours after you add a germination pad in there. And if you see an increase in signal or the line gets darker compared to that baseline test, that means you still have either one of 2 things. There's active spores that, again, need to be monitored regularly and treated if a humidity or the right environmentals happen, or you still have an active infection and maybe you have some resistance built up to the fungicide you're using. Maybe it was inadequate coverage, or maybe it just didn't work this time. You missed that spot. So again, you want to take a baseline and then run it again so that you can see the increase in infection, if any. This gives you the ability to either break up your schedule, maybe delay sprays, work with your agronomist on this and make sure you're not exposing yourself to excess risk. But if it's not present, you don't need to take action on it. So this result gives you, a good read on if you were effective or not in your treatments. So the next one is in post-harvest. So we have had some really cool results using this. You can set up a different protocol, which we're happy to help, and we calibrate your testing protocols with a reading on this cube reader. But it's quite simple. You take your samples off of your finished product after harvest. You run a series of immediate tests, and that tells you the infection level in your harvested crop. And with those quantified results, with strawberries or grapes, some of these fruit products, it's a percentage of infection before you have to hit a certain percentage of infection to be eligible to go into cold storage or shipping. So there's a lot of groups, shipping, especially South America, Australia, all across the globe, people are shipping fruit internationally, and knowing the level of infection is absolutely critical for that long-term transport. Maybe you need to airlift them across overseas, or maybe you're okay to put them in a shipping container. So very important step. And with grapes, we have it very well dialed. You take 1,000 berries from the field. They have to test for sugar and coloration anyways. So all you have to do is you crush those grapes after you've collected them. We provide a pipette. You take a certain amount, add the grape juice into the test tube, and run the result. And we can quantify from 0.5% all the way up into the 20%. Last year, we were on site in a farm in the Central Valley of California, and we did just that. We took some samples as they were harvesting and packaging. Unfortunately for them, when we got the test result, we went home, ran the tests, and we showed about a 15% hidden latent infection. When they took that to cold storage, surface sterilized everything, got it all packed and put into the freezers, and they still lost their product because they had a 15% infection. Cost about $1 million in crop loss for just that one farm. So knowing before you even harvest or post-harvest into the supply chain really can make a difference in ensuring that your product gets to the end customer and you get a top price for your product. And again, we're more than happy to help set up these protocols and do the work to calibrate for your use setting. So whether you're in strawberries, grapes, any other crop, we can easily set this up so you have quantifiable results before shipping off into the marketplace. So any questions, follow-up, or if you'd like any support with these protocols and setting up a test or even interpreting a test and seeing what the test means, feel free to give us a call or send us an email at any time at hello@cubelabs.com or visit us at cubelabs.com, and we'd be more than happy to get on a call with you and speak